Hi, everybody. Hi. If you don't already know me, I'm Natalie, and it is my absolute pleasure to introduce Dr. Marlene Wolfe, who is an assistant professor in environmental health here at Emory. As an environmental microbiologist, engineer, and epidemiologist, Dr. Wolfe's work focuses on developing tools to use environmental detection of pathogens to understand population health and risks, and to implement and evaluate interventions that reduce environmental exposure to pathogens. She uses mixed methods approaches to understand the efficacy of interventions in the lab and how they're utilized and effective in context. Her work particularly addresses complex issues in water sanitation and hygiene, and infectious disease in communities that are low resource or facing emergencies to create sustainable and adaptable solutions to understand and manage our relationship to infectious disease in our environment. Take it away, Dr. Wolfe.
Thank you. Hey, hi everybody, and everybody online too. It's great to be here, and I've got a lot of slides I'm going to go through and talk about a lot of things, and I'll probably go fairly quickly so that we have plenty of time for conversation and questions. But you know, you can stop me if there's something that's confusing, and if I go through something quickly and we want to kind of go back to it and talk about it a bit in conversation, that would be great. But I'm going to generally today focus on talking about one area of my work, which is a pretty big area, something I'm spending a lot of time on right now, which is wastewater monitoring for infectious diseases. So really thinking about wastewater monitoring as a nimble approach that we can use to address a very diverse set of outbreak disease threats. This is a little of my personal history, and I can talk more about how some of my different experiences have led me to where I am today, which is being a sewer person, and I never really thought my career would turn out this way. It has given me zero legs to stand on when my kids make poop jokes. I'm like, I can't stop you. I have just like, you know, no legitimacy there. So I have to hand that parenting to my husband, but it is, I feel one of these things where my career looks pretty linear if you look at it. But in my experience and in the process, no part of it has felt like the next step is kind of the obvious follow-up of the one before it necessarily. So I think that's something important to remember, especially because I feel like I am a person who has been doing, in many ways, kind of the same thing that I started doing when I was 19 and I had my first research and field experiences. But in reality and in my personal experience, you know, it has not felt simple and straightforward. So I did my undergrad at Stanford. I majored in human biology. This is my first field and lab experience in Tanzania when I was 19, and I left the country for the first time and went on a pretty exciting and extremely arduous adventure of doing some work on water quality and hand washing with some researchers in Tanzania. And that was my first field experience. It was incredibly challenging. We worked really long hours. We worked really hard. I didn't get to see a single interesting thing like I didn't go on safari, I didn't do anything. I like worked in an apartment that we built in our lab, and I was just like completely addicted to it. I loved it. And so it was sort of like, well, if you like that, then this might be the field for you, because that wasn't supposed to be fun. So I got into research in that way, and then ended up, after graduating from college, spending a couple of years living in Kenya and working on a large field trial of water sanitation and hygiene. So a lot of my work was really WASH and low and middle income countries focused. I did a master's in epidemiology and then a PhD in environmental health engineering at Tufts. And those actually kind of overlapped a little bit. I have two kids again, so poop jokes all the time. I have two kids and had both of them actually like within my PhD program and just as I was finishing, which I like to mention because I think that that's something that we don't talk about very much, about how you kind of fit some of your family and life into your career as well as your career. And in many ways, having my kids during my PhD was a great time to do it. And in many ways, it was very challenging. So I finished up my PhD and was really focused on laboratory efficacy experiments and still a lot of work on WASH and low and middle income countries, and went and did a postdoc at Stanford. And during that time, there was a little pandemic. And so I shifted a little bit from some of the international work that I had been doing and started doing more domestic work, including work on wastewater monitoring, starting out with COVID and then expanding to a whole slew of other things. I still am doing some work on hand hygiene, some work on interventions in low and middle income countries and WASH, but a lot of my work is now on wastewater monitoring, both in high income and low income settings. And another area that I've kind of expanded into is I've done a lot of work lately on advocacy and policy related stuff. So this is my personal excitement of the recent past is this picture. I got to go to the White House last week to talk to people about wastewater monitoring in the same session as people who were talking about satellites and drones. And then I was again in like the sewer person section of the room. And I've just learned to embrace that. And it's just as exciting as satellites. And it just as much got me into this very exciting conversation at the White House as it did, the people who are working on satellites. So it's exciting to be in a position where I'm doing a lot of work with CDC and a lot of work with others in state, local, and also federal government to think about how do we support our infectious disease surveillance systems, prevent health emergencies, prevent outbreaks and pandemics. So that's my, personal history. And I'm happy to talk about, career and all those things, but I want to give you a bunch of information about wastewater now. Okay, so wastewater. How many of you have heard about wastewater monitoring for infectious disease before? I hope it's quite a few people at this point. Awesome. Okay, cool. This is nice because it's not like a brand new thing anymore. Some of you might have heard me talk about it before, but it's also kind of a field that has grown a lot in the past few years. It's something that's been around for a long time, but has really experienced a renaissance recently. So we've seen really big changes in the abilities that we have in this field and the utility of it. So the basic premise is that we can get insights at a community level about infectious disease because when a person is infected with an infectious disease, they're going to shed it in their bodily secretions, right? Like that's kind of how infectious diseases spread a lot of the time, right? So it's understandable, but there are also a lot of ways that we shed viruses, bacteria, fungi, their components in parts of our bodily secretions that maybe aren't even associated with onward transmission. And so wastewater, then if it's capturing everything that goes down your toilet, your shower, your sink, your washing machine, everything is going down the drain and into the sewer. And that provides a composite biological sample that can provide information about the whole community in this really accessible one single sample from a source. And so there are some advantages to this. This can promote health equity because we're including everybody who's connected to the sewer system, regardless of what their access is to healthcare services. Now, the obvious flip side of that is you do have to be connected to the sewer system, right, to be included, but 85% of Americans live on networked sewer systems. And so that's pretty widely inclusive if you think about what are our different systems and what percentage of people do they include in the United States. 85% is pretty good. And there are also a lot of ways that people who might be on septic or in communities that don't have sewer networks, they are traveling into town for work and they are going to the bathroom there and they're kind of represented in different ways. So it's a very inclusive process to get information. And it provides these community insights, especially when clinical data or other sources of data is really incomplete, slow, unavailable. You think back to the beginning of COVID when nobody could get a test. You think about the beginning of the mpox outbreak a couple of years ago when people didn't necessarily know what they were looking for, and a lot of communities that were heavily impacted didn't have very good access to healthcare, and there's a lot of stigma associated. So there are a lot of different ways that we can get that good information that's anonymous at population level pretty quickly when other systems might be impacted or difficult to use. The basic testing process is that we're typically using molecular methods to target genetic material of the pathogens of interest. So we're not usually culturing these bacteria or viruses to look for viable pathogens that are going to be passed on. This is really an indicator of what people are infected with. So sample collection happens very often at the utility level where wastewater is treated before that water is discharged back into the environment. And so this is an aerial view of a very large wastewater treatment plant. We'll collect those samples as they come into the utility. So they haven't experienced any treatment yet, and they're full of many, many things, including all of the diseases that we might want to target. The sample's prepared and the nucleic acids are extracted, and then we do detection and quantification using thing that I use most in my group is droplet digital PCR to look for particular regions of the genome that are unique to that pathogen, to detect it and quantify how much of it is present in the sample. We also do sequencing to help characterize the sample. So we do a lot of sequencing for COVID, for example, to look at what different SARS-CoV-2 variants are represented in wastewater. And then we analyze the data and share it. And so this is an example I like to show because the blue here, you can see this is a timeline of, this is early 2021. And the blue is wastewater concentrations of SARS-CoV-2 over time. And then the red is the 7-day average of COVID cases per 100,000 people. And what you see here in the beginning of the year, this is in Coeur d'Alene, Idaho. And you see that the cases track really closely with this significant peak from the winter coming down into the spring. They match up really nicely. But then you'll notice that the wastewater goes up to pretty high levels again, while the cases go up, yes, but they're not really seeing that magnitude that is at all comparable to what was happening earlier in the year. And, you know, why might that be? Does anybody have an idea? Why do we see that like shift?
The variant that was transmitting at that time was like less severe, so people wouldn't have their case reported in surveillance systems.
Yes, so the second part of what you're saying, the people might not have their case reported. That's very true. And that is basically what's happening here. But most likely the main reason for that is because at this time period, right in April, is when government subsidized testing ended. So it wasn't even actually having to do, it would be a good guess that like maybe something happened with severity, so less people were presenting or needing care or needing a test. But that actually probably isn't even the reason. It's just about availability and the cost of testing. And yeah, we see an increase here. So anybody looking at just the case data at that time could go, oh, there's a bump in COVID. Okay, good information to have. But when we also have the context of the wastewater data, we can say it's likely that that peak here was much closer to that winter surge than you would think if you had just been going off of this because the reporting practices changed so much. And that's continued to happen over the years, that these significant changes in how we measure and report and handle COVID have adjusted what we are, you know, the way that we can ascertain what's happening with the outbreaks. Wastewater, we definitely can have changes. You know, different variants might be shed in different quantities by people who are infected, severity, vaccines, all kinds of things could have an impact on that. However, in reality, we've seen at this kind of aggregated population level that things have stayed very consistent over time. And so there are much bigger changes happening in the testing practices. So you can see how this data has become really valuable to give us a clear sense of context and comparison for COVID. The other really wonderful thing about this testing process is that in many, many cases, the same process can be used to collect and concentrate a sample that can then be tested for a lot of different things. So we can do this detection and quantification with digital PCR on one sample that's collected and prepared, and then we can actually multiplex, run several tests at the same time. And from that same sample that is about like a pea-sized amount of wastewater solids from the utility, we can get information about many, many different targets that represents anywhere from 10,000 peoples, like the smallest utility that we work with. We actually put that as a minimum because we want to maintain anonymity of people who are represented in the sample. Then our largest utilities we work with serve over 4 million people. And so we can just get incredible information on large populations with this tiny, tiny little piece of waste, which we don't waste. Okay, so, but what if you don't have a sewer? Yes.
So, based on what you just said, like how frequently do you test the different, I guess, sewage plants that serve different communities? Like, do you do the larger one first and then scale back to do the smaller ones? Do you start with the smaller one first and like?
Yeah. That's a really good question. I'll talk a little bit more in a minute about how like our program runs and how we do that testing. But one thing that has been heavily debated and has a lot to do with funding is, you know, how much do we sort of look at the large scale versus then drill down and say, okay, well, I've got something going on in this area, you know, our treatment plant in Los Angeles that serves 4 million people. But if I see dengue in that, you know, plant that serves 4 million people, how can I get a little more specific about maybe where that's coming from? And there are projects that we've done to look at smaller areas of the large area that's serviced by a treatment plant. And the biggest challenge with that is the funding to be adaptive to collect those samples. If they're not coming from the utility, it's quite a bit more expensive for us to have staff go around and pop manhole covers and go out to places where we can get those samples. But we can get, you know, speaking about across scales, right, we can get really valuable information about, you know, going upstream to help identify a little bit closer where is the real problem within a community. The other issue is that because this is a passively contributed anonymous sample, we want to be really careful about how much are we narrowing in to preserve that anonymity. And there's been a lot of debates in the field about what is ethical and what's appropriate in terms of you know, there's one person who I can think of who's done some work that really kind of tracked down an individual super shedder. And in my opinion, I think that is not ethical. You know, that person did not consent to be tracked down through the sewer. So we have to be careful about that. You know, this is population level data, IRB doesn't apply, but that doesn't mean there aren't ethical guidelines for us to adhere to. So we want to balance some of those considerations and the cost considerations with the possible value of going more upstream in these networks to get more specific information about outbreaks. But yeah, that's a really good question. And I'll talk a little bit more about how like our program is structured. So what if you don't have a sewer? There is, you know, the broader field is environmental surveillance, and it includes monitoring outside of design network sewer systems. There is a lot of work that has been done in this way. A lot of environmental and wastewater monitoring has been done over the past several decades for polio in endemic areas of the world to identify outbreaks, and that has a lot been based on environmental samples that are not from networked sewer systems. So this can be a little trickier for communities that are not on network sewers, because we do want to find something that's representative of a population level and not just, you know, going to someone's individual latrine and picking out a sample. That's harder than sticking a swab up their nose or doing some other sample collection, right? We want to look for these areas where we've got kind of population level contributions coming together, but there are a lot of places that that's the case, and we just have to kind of contextualize it differently if we're not talking about a networked sewer system. So we have a incoming PhD student this year in EHS, Brandy, who has worked on wastewater monitoring in a refugee camp in Malawi that she was working in this past year as part of her job. And so she's done a lot of interesting work thinking about getting these samples and then how we can interpret the data from them. So there's a lot of opportunities outside of network sewers in the US, but that's kind of what I'm going to focus on for right now. This is that history of wastewater monitoring that I was mentioning. So the sewage used to be used to monitor enteric pathogens, especially polio in endemic areas. Really had this shift during COVID to say, wow, this respiratory virus is shed a lot in feces. I bet we can track that in wastewater, and kind of cracked open the can of worms of what are all the other things we can monitor in wastewater. And so now the expansion of monitoring things in sewage has gone very, very wide. And so there are so many more things that we're doing now, partly because our technologies to do that molecular testing have improved and given us a lot more opportunities. Some of the priority areas that we look at are diseases where there are significant surveillance gaps. So that's true for many respiratory diseases, diseases that are vaccine preventable, so rotavirus or measles, you know, where there might be some vaccination campaign action that could be taken with information from wastewater. Diseases that are rare and high consequence, where we're really kind of initially looking for the presence of, you know, detection in wastewater at all would be really meaningful. So Mpox, Candida auris, some future disease X that we are concerned about. And then diseases where maybe we don't have as much information as we'd like about the underlying epidemiology. So norovirus is a good example of where we could learn more about the epidemiology of the disease, because most people are not presenting to a healthcare provider in a way that's giving us a lot of information about their condition or about the particular infection that they have. And then who's involved in wastewater surveillance? There are a lot of, I'm gonna try to go through this really fast, but there are a lot of different entities that are involved. So the federal government, CDC has a national effort to fund, coordinate, provide guidance for wastewater monitoring. It's called the National Wastewater Surveillance System, NEWS. And so, we work closely with them, but they support states to do wastewater monitoring, and then they provide guidance and funding, and they work with that data. So states are kind of the main entity that are responsible for public health in the United States, but also specifically for wastewater monitoring. So they receive resources and guidance from CDC to implement programs, and then they share that data with CDC. Private industry, so there are companies that run from having full service operations to do wastewater monitoring. CDC has a contract with a company that does wastewater monitoring for them to kind of fill in the gaps from states. And then groups that maybe do sampling or produce some specific testing products. There's been a proliferation of products that you can get that are specific to wastewater surveillance now. There are a number of nonprofits that provide resources, and then academics have had a really important role in leading wastewater monitoring because this still is a really new and emerging technology in a lot of ways. And so there's been a very interesting interplay that we have had with the federal and state governments who are doing this work and implementing it, because their money and their resources are designed for practice, for public health practice and implementation. But a lot of the things that we're doing are very new, and they're very new science. And so as researchers, we have the opportunity and responsibility to develop these things, test them out, whether they're going to work or not. And then if they work, they will be adopted. And so we've seen that. I'll give a couple examples, but we've seen that happen repeatedly where we've kind of innovated on things in a research setting in partnership with these practitioners, and then seen that get picked up by these programs to become a part of practice. I'm going to skip through these individual slides on each of these because it doesn't add much, but I'm happy to share these with anyone who's interested. Okay, so then this is the program that I co-lead with my colleague at Stanford, and it's called Wastewater Scan. We have 148 wastewater utilities across the United States. It's about 12% of the US population that's represented by those sites, and they're in 40 states. And the goal of this is basically to scale an academic program, again, this research and based on innovation, to give us this platform to innovate on methods for sensitive detection of these priority diseases, develop data insights for disease epidemiology and outbreak response, and work on translational science to support departments of public health. So we have close partnerships. I have spent a lot of time talking to people from most of these sites all across the country. It's been a significant part of my time developing these relationships with so many people from sites, but especially we've had close partnerships with Georgia and California DPHs because that's where each of us are based. And so it has been this opportunity to have a program that we're running on a regular basis, and we have contracted a company. We've been working with them for 4 years now. We've contracted them to help us power this program so we don't have students and postdocs running all the samples from this program, but we have it running consistently and work in partnership with all of these sites to deliver data on a regular basis. They submit samples 3 times a week and work with them so that if we go, aha, there's a lot of dengue in Miami, we'd really like to do a project on dengue, and we have 3 sites in Miami, let's call them up and see if they want to work with us on that at the same time as we're giving them regular information about COVID and influenza and RSV, or oh, there's a human case of H5N1 in Amarillo, Texas. We have 2 sites in Amarillo, Texas. Let's call them up and see if they want to work with us on this. And so it gives us this platform to do relevant research in real time that then we can feed back to public health departments to actually be utilized, and we can take from them what they're going to teach us about what they need so that we can develop tools that are actually useful. I'm going to skip over SARS-CoV-2 because I already kind of talked about this example, but this is just to show that we can characterize variants. This is sequencing data from wastewater. This is from yesterday. So this is our recent surge nationally. This is across all of those 148 sites. We're coming down off this latest peak, which is good to see, but it's not been good. So again, one of the best sources of information I think we have about what's going on with COVID right now is this wastewater data. And these are all the targets that we're running as part of the wastewater scan program right now. So we have these respiratory viruses, SARS-CoV-2, influenza. We're looking at influenza A and B. And I guess I forgot to add this. So we're looking at H5, bird flu, but then we're also looking at H1 and H3 to give us information about all the different flu subtypes that may be circulating. RSV, human metapneumovirus, enterovirus D68, which we've actually been seeing a lot of lately. Norovirus and rotavirus are our current enteric viruses. And then these other fun candy bag of pathogens, Mpox, hepatitis A, and Candida auris. So we're running all of these on every single sample that we get. And then we're also doing kind of additional, like I said, research to figure out what are some other things that we can monitor in wastewater. So here are some examples of things that we've done, the innovating and then the scaling on. So influenza is here on the left, and you can see this was an initial study we did. There was an outbreak investigation that was done at University of Michigan. And so this is the case data from that outbreak investigation. And then this is the wastewater data on influenza from that same time. So that was our first proof of concept that we were seeing this close relationship between the influenza and wastewater and the number of cases in the community. And then we were also able to show that we're seeing sort of a peak in hospitalization rate nationally, which is in red here, at about the same time within a couple days of when we saw the peak in wastewater at that national level data. So useful for monitoring influenza season. RSV, very similar. So we were looking at here, this is the wastewater data. Copies per gram of RSV and wastewater, and then this was the positivity rate across the state for that same time period. And so you can see that the wastewater measurements reflect these measures of disease in the community. And then Mpox was very interesting, a little bit different one, because there were a lot of questions about whether this virus could be observed in wastewater. So it's really different from some of our respiratory and enteric viruses. But we've had good success with monitoring Mpox in wastewater. Been having a lot of conversations in the past couple of weeks with the sort of new outbreaks of Clade 1B Mpox that are spreading globally, kind of. And so, like, needing to look out for that. But what you see on the top here is, again, the wastewater measurements at these 3 sites, and then the number of cases that were reported at those 3 sites in the same time period. So amazingly diverse set of things that we can monitor through wastewater. And then there are other, so we've added, all 3 of these things. We're just on that slide of stuff that we're running every sample, right? So it's part of the regular program. There are other things that we're innovating in the early stage. We're like, let's see if we proof of concept, might this work? And then what should we do with that data? How could it really be useful for public health? So a couple examples are HIV and dengue. So HIV, we've got, paper submitted on this. And we looked at 2 years of samples from the Bay Area, and we saw a difference in sort of the rate of detection and the concentrations of HIV nucleic acids in an area with a higher prevalence of people living with HIV compared to an area with a lower prevalence of people living with HIV. So still really working on what are the use cases potentially for this. There are a lot of groups involved in that conversation about how might you use this kind of data to contribute to, ending the HIV pandemic. So that's one area of ongoing research that we're not incorporating into regular monitoring right now, because it's not at that point, right? Dengue is closer to that point. So this is some of the work that we did in Miami to detect dengue RNA in wastewater. We have kind of expanded that to a couple areas, other areas around the country where we're starting to do more regular monitoring data, and CDC has also been kind of interested in continuing expanding more of that regular monitoring of dengue in wastewater. Dengue is shed in urine if you're infected with dengue. And as we see climate change impact where dengue may spread within the United States and globally, this is a great opportunity for us to have our finger on the pulse of where is dengue right now. So these are some of the new things that are coming up. I'm just going to go through a couple short things more. this is our process for developing new targets. I'm like throwing around all of these things that we're monitoring, and it seems like a very grab bag, random list, right? We do have this process that we go through to develop new targets and add them into the program. And that involves taking a lot of input from our public health partners and others on what would you like more data on, right? What would be helpful? You know, what else can we provide? Then looking at, you know, is there a technical feasibility of testing for that? Is there an assay that we can find or that we can design to use and apply to wastewater? And then also, is it shed into wastewater? Like, is that very likely? And then if we've met all of those criteria, then we'll kind of find some target sequences that we're identifying. Right now, we're going through this with mpox clade 1. It's actually really tough. There are not a lot of sequences out there for the new clade 1b cases, which have some substantial mutations that make some of our current assays not work very well. So we need this good information about the sequences from current outbreaks to be able to design assays. Then we'll procure the materials, test specificity and sensitivity to make sure we're really picking up exactly what we intend in wastewater and that we're very sensitive to do so. We'll apply that assay to a set of samples where we think there's some potential for detection, compare it to relevant clinical data. And then if that all looks like it's matching up pretty well, we'll pilot the testing with some of these partners we have all across the country. And then if it looks like it's working well and it's something that is adding value to the public health departments that we work with, then we'll actually add it into the program. That is the process that we go through. This is just more in depth on the pieces that I already talked about. And the last thing I want to mention is bird flu. So, this year there has been a big outbreak of bird flu in cattle, and a spillover event likely introduced the virus into dairy cattle in late 2023. Dairy from cows is much more likely to end up in wastewater systems than some of the contributions from poultry or wild birds, where H5N1 has spread in bird populations, primarily in the United States over the past few years. So there was this new concern about, wow, we might be able to identify this in wastewater. H5N1 infections in humans have been very serious in previous outbreaks. There have been several cases now in humans in the United States in this latest outbreak that are spillover cases, where animals have transmitted the virus directly to humans. And so this seemed like a good opportunity for us to potentially look for this in wastewater. So again, we reached out to a bunch of our partners. I'm not gonna delay on this data here right now. We could come back and look at it. But the main takeaway that I want to share is that we were able to reach out to these partners in locations all across the country where there were outbreaks in cattle and where there were not. So in Gilroy, California, and in Honolulu, Hawaii, there were not outbreaks expected. And the blue is just influenza A overall. So that could include any influenza. And then the red is H5 influenza. And so you can see in these 4 sites where we did have nearby cattle outbreaks, we saw the H5 marker showing up in wastewater about a month before the outbreaks were detected in cattle. And in the places where we didn't have cattle outbreaks, we did not see any detection in wastewater. So it's a very kind of unusual application because this is a virus that has primarily been infecting animals, but we have this really great opportunity and challenge to detect it in wastewater. I'm going to skip through some of the details. We've rolled this out now, so all of the sites in the program are getting that testing for H5. A little controversial, little bit of many conversations with lots of people about this, but it has been really valuable information to give context about these outbreaks. And we are sharing all of the data on our website. So as soon as the data is available for all of these things that we run regularly, we put it on our dashboard and it's available to everybody. And so we've spent a lot of work building out this dashboard to enable people to explore, play with the data, generate insights, and just kind of with the belief that it is important for us to make this data accessible to people right away, rather than kind of putting it behind a password or only providing it to public health officials. So it is available to everybody. And we have social media where we post about it and post insights regularly. So follow us on Instagram, because our team spends a lot of time putting together updates on what's going on with all this data there. So takeaways, we can provide rapid, valuable data on occurrence and outbreak characteristics for many diseases with wastewater. Our public health and academic partnerships provide opportunities to quickly respond to new things and implement wastewater testing. A structured process of choosing and developing new targets is important because we want everything to have a strong evidence base and be trusted data. And these close partnerships with diverse, widespread, and ideally we can get more and more strategic in terms of representation in our network, support the development of these widely applicable data insights and models that we can develop with the help of all of our partners. So that's it, and I'm happy to talk more and answer some questions.
So I have two questions. What have been some of the barriers for establishing the wastewater scan in certain states? Because I saw like South Carolina wasn't included. And then my second question is, in areas that have a more dominant agricultural component, do you also scan for animal pathogens that would serve those communities as well?
Yeah, good questions. So one of the challenges with this field, not just for us as academics, but I think for everybody, is that there is really a separation between wastewater treatment utilities and public health departments and researchers who are working on public health, right? I mean, while it is a very important public health service to make sure that waste is diverted, right? These two, groups of, government workers are not very connected. And so there isn't, public health departments can't say, hey, we need a sample from there, hand it over. They're completely separate. So we're relying very much on the goodwill of people who work at wastewater utilities to spend extra time getting a sample, packaging it up and sending it, and they're not compensated for that. We actually try to provide some compensation to offset the cost of the labor. It's the type of work that is part of their normal activity, so they're trained to do it, they know how to do it, they have the resources, but many, especially smaller utilities or rural utilities, will have one person who's working there. And for them to go around and grab that sample and send it to us is a significant added burden, and no one currently can compel them to participate. So we need sort of the cooperation and the resources to make it like really realistic for us to get these samples from all the places where we would like to have them, because it's a small percentage of utilities across the US right now that are represented. The other challenge is just funding. And so for us, that wastewater scan network, I mean, it's very much a convenient sample. We've tried to be strategic. We want to try and go and say, hey, you serve a really important population. You're in a really unique position. You're, an important port of entry into the US, or, you have a lot of people in your, network who are traditionally underserved by other systems. And so we want to make sure you're included. But as much as we can go make that pitch to people, they don't always have the resources to participate. So that's something I think we're continuing to work on and that people are thinking about at a lot of levels. How do we get the strategic networks and the more complete coverage that we would really like to have? And our program is a very small subset of, several thousand sites that are doing wastewater monitoring across the country right now, as part of state programs and other things. What's your second question?
Oh, so for states or communities that have a strong agricultural component.
Yes.
Do you alter the screenings that you?
We don't. So we are doing the same testing everywhere. There's been conversations about, well, maybe we could do one set of things everywhere and then like another panel that you run on some places. And then, if there's a health emergency, you run that panel more places. There's ways that the system could maybe be adapted. That requires funding, I think, is the main challenge. We don't monitor for animal pathogens for the most part. The bird flu outbreak has been a very different thing. And the main reason for that is because municipal wastewater is primarily a human sample. So most systems are not combined. They're not accepting stormwater and other inputs. They're coming from buildings, businesses, they're human inputs. And now some people toilet train their cat, right? So, I mean, it is possible that you have other inputs, and we know that there's milk going down the drain. Trying to remember the statistic, because Abby, a postdoc in my group, was just writing this up in a paper about what percentage of milk at schools is dumped down the drain. So everybody gets a milk at lunch. And it's a lot. It's like a lot of milk. And so that's why we're seeing bird flu so much. It's one of the big reasons. but it's not a reliable source for most of our animal associated output, but that can change any minute, just like it did with bird flu, so we're open.
I think I saw on the slide where you listed the pathogens you were testing, some control samples that you were getting. I was wondering if you need to include like some sort of baseline metric to be able to delineate what are increases in, say, like a pathogen in the population, or were just like increases in input into the wastewater management system.
Yeah, that's a very good question, and partly what our controls are for. So there's two, and they're listed here. One is pepper mild mottle virus. That's an endogenous control. It's a virus that infects peppers, and it is very prevalent in wastewater. And the main way that it's entering wastewater is in the feces of people who eat peppers. And so it helps serve as a little bit of a control for the fecal input into the sample. So we use that as one method. Bovine coronavirus, we spike that into all of the samples, and then it's a process control. So we look to make sure that that was extracted efficiently and in the quantities that we would expect, so that we know that the process was working. The other thing is that we express all of our results in copies per gram dry weight of solids. So we're looking for these targets in the solid portion of wastewater. We settle out the solids and use those. And then we take a little piece of that and we dry it down so that we have a measurement of how much actual like solid matter was in that sample. And so if we express then the results in terms of the solid matter that was in the original sample, that controls for things like dilution events when you might have much more water flowing through the system one day than another. So we have a few different ways that we control for that. But yeah, very important to understand the results in context of the inputs. Yeah.
I had a question about the dengue surveillance in Miami. So I have two kind of related questions. So the first is, was there monitoring and sequencing of mosquito samples in the area? And if so, were you seeing similar distributions of the serotypes and mosquito populations versus like actual clinical cases versus wastewater surveillance?
Yeah, so what's interesting is that during the time period when we were doing this testing, there weren't any mosquitoes that tested positive. So that ongoing surveillance was happening. A lot of people spending a lot of time catching a lot of mosquitoes, and there weren't any that tested positive. So it's, complementary surveillance stacked together to help understand what's going on. We can't through wastewater distinguish between locally acquired cases from a mosquito that was, present in Miami versus people who just, traveled back from places where dengue is endemic. So we can kind of put these pieces of information together and say, what is each lacking? But it was interesting that there were a lot of mosquitoes captured and none of them were positive for dengue. And there were a number of cases within the area that were detected and that were suspected to be local transmission based on the travel history of the individuals and other contextual information. So we did find that there were, the bottom plot here is the reported cases of dengue in Miami. And then the top here is the different colors of the different sites. We had 3 different sites. The detections of dengue and wastewater. All of what we detected in Miami was serotype 3. And under here, you can see this is a combination. I didn't put the legend on here, so that's a little confusing. But the light blue is like serotype undetermined and the dark blue is serotype 3. So we were really seeing that during this period when there were a bunch of cases, there was consistent detections in wastewater. But it is those cases that we're seeing of dengue are very much expected to be the tip of the iceberg, because the presentation, especially of a first case of dengue, is so similar to a lot of things. It's not necessarily identified in a clinical setting. So yeah, I mean, we saw about 1 detection limit was around 1 case reported per million people. But again, if there's 1 case per million reported, we expect that there are many more that are unreported. No mosquitoes.
Awesome. Yeah, thank you. Can I ask one more question just about the assay development design? So I know it's going to differ depending on what pathogen you're looking at, but what's your typical like lower limit of detection?
Yeah, it really depends. And part of that is dependent on us having like really good information about the cases. So we actually know that we're capturing cases. So our best information about that comes from when we were counting COVID cases really, really well. And what we were seeing was that it was between 3 to 5 cases per 100,000 people was our detection limit. So, very few cases in a large population, and we suspect that our detection limit for some of our respiratory viruses is very similar. It's a lot harder for us to determine what that is likely to be in terms of number of cases in the community for something like dengue, where we just don't really have a good sense of actually how many cases are there if we're detecting, 1 in a million people. It's probably more, especially if there's local transmission.
Yeah, great talk. Thanks so much. I just had a question, or I wonder if you can talk a little bit about like phasing of mutations. I know that was like a huge thing early on when wastewater was kind of getting off the ground. People were concerned about SARS-CoV-2 variant tracking, and I just like have no clue kind of like what has happened since then. So I assume for like SARS-CoV-2, this is based on like S protein, and it seems like for the flu stuff, you're using like M protein or H5. Yeah, can you just like talk a little bit about like what's happening there?
Yeah, so we were for a long time doing variant detection that was based on doing PCR for characteristic mutations of the variants. And so those were, could be in a number of places on the genome. This is, if you look at the match between kind of up here, this is the wastewater data. We actually kind of name the mutations that were targeted here, and then they go with these different variants that are down here, and you can see how the, proportion in clinical cases tracks really closely with the proportion of what we're measuring in wastewater, looking at that mutation, and then comparing it to the concentration of conserved region of the N gene. When we started doing this, we were doing an assay on the N gene, S, and ORF1A, and we were measuring all of them, and they all looked exactly the same. So then we just went down to doing just the N gene. We have kept up over time with making sure we don't have any mutations in that region, of course, that would cause dropout. That did happen with the S gene assay, which we dropped anyway because it was redundant at some point. But that's one way. And then the other way is that we've done sequencing. So we're able to do tiled amplicon sequencing of SARS-CoV-2 RNA in wastewater and looking at the proportion of reads that are aligning with these different, known variants. We can come up with the kind of proportion in wastewater likely of, kind of different variants. And that has lined up really well with the proportion of clinical cases sequenced that are assigned to each of those variants. So right now we're doing this, and we have a subset of our sites, around 30, that we do regular sequencing on. So this plot is from our website.
I was wondering if you had some pathogens that are on like your wish list, or if you have more funding, like what else would you want to surveil? Because I understand there's going to be limitations, like, it has to be excreted in either urine or feces for you to pick it up. But
Yeah.
I thought it's interesting that it sort of says that you just have viruses and then like one fungus, or is there any reason why?
Yeah, that's very true. It's a good question. Viruses are great because they're pretty straightforward. They are not multiplying in the environment. They are there's their genomes are simpler. There's a lot of things that make viruses a little bit easier to handle in these environmental samples. There are fewer things in the wastewater and environmental samples that are similar to the viruses than there are to many pathogenic bacteria, and so they can be like a little easier to pick out. So there's also a lot of our sort of emerging outbreak threats are viral, and so kind of put those two things together, and we have focused a lot on viruses. Candida auris is a fungus, so that's the oddball in the list. We're working on a number of new things right now that include bacteria. So that includes like pertussis, we're looking at tuberculosis, we're looking at, trying to remember everything that's on the list right now because we're just starting up the project looking at some STIs. So that we are getting more into that. And so that is going to be exciting to have those methods working well and hopefully working like right alongside some of the viral targets so that we can use the same or very similar process. We've done some work on antimicrobial resistance targets in wastewater. So Maya Non-Poly here in environmental health, she's done a lot of work on AMR, and then I'm kind of working with another group where we're also doing some work on those targets. But the data can be difficult to interpret because you're looking at, in some cases, at resistance genes, but knowing what organism they're in and if they are pathogenic and if they're just in the environment or if they came from people. There's like a lot of things that's complicated in the interpretation. So I'd say that's one of our like big developing areas. Yeah, it's a gap right now.
I have another question, if that's okay.
Oh, yeah, you have, sorry, I just saw you asked a question yet, and then I'll back.
I know these are obviously owned by, the federal or state government, but have you done any work with like maybe prisons or like any sort of congregate setting for nursing homes? Or does their wastewater go to the same place, or I just was curious?
Yeah, so their wastewater does usually go to the same place, so like ultimately ends up at the utility. We have done some work with Fulton County Jail. So I worked with Ann Spalding in Epi. She does a lot of work with the jail. And so she had a project that she did that was focused on looking at COVID, and they were doing tests and comparing that to wastewater data and seeing like what's the utility. But we've also done this project where we were looking at actually doing testing directly from the jail for Mpox during the outbreak. It's a big concern. But then also talking to people with lived experience of incarceration and talking to workers at the jail also about their perspectives about doing testing in that kind of setting, because you get into a lot of tricky territory, right? Because on the one hand, when people are incarcerated and they are, held somewhere involuntarily, having systems to understand the health concerns and respond to them quickly is especially important because there's that like extra imperative to do so. However, people also are in relatively small setting and they don't have a choice to kind of opt in or out of that testing. And so there's a variety of opinions. We're working on kind of bringing this research together and writing it up right now about like, well, do people want that? Do they feel like that's helping to protect their health, or do they feel surveilled, and like how could results from this kind of testing be expressed in a way that is supportive to the health of residents in these facilities? There's also work that's been done on like nursing homes and such, which is a really different area of concern, but has a lot of similarities in terms of the actual monitoring and testing. To me, a big challenge and what I have really not liked about doing the work with the jail is that we can provide data about what's going on, and we can provide data showing there's a problem. But if nobody does anything about it, then there's nothing I can do, right? There's nothing I can do to make Fulton County Jail respond to the health needs of people who are clearly sick on their watch. And so at what point are you saying, well, I'm participating in a system that is fundamentally unjust and like fun? So like, do we want to participate to help contribute to better care? Or do we want to say, well, it looks like they're not picking up what we're laying down and they're not going to respond to it no matter what? Maybe this isn't the place for us to be in partnership. I think that's a very tricky conversation, and there are a lot of different, like, very valid opinions about what the right thing is to do there. We have done some of that work, and I think it's been very complicated, including the day when Trump was being arraigned at the jail, and some of our staff had to go down to pick up samples, and I was like, well, let's maybe skip this day and not go down there right now. So it's complicated, right? But there's potential, but it's so tricky to do the right thing for people in an unjust system, I think is what it comes down to for me. You have a question?
Yeah, so I had a question about the relationship between like shedding of enteric, also non-enteric pathogens, and then what's known about the period of time that those people are infectious. I feel like some of the motivation for wastewater testing is that you maybe get this like early warning system, which obviously works if people are shedding before perhaps they're infectious. Is that the case for SARS-CoV-2, and then is there information about that for some of the other pathogens that you're working with?
Yeah, it appears to be the case, although you can imagine it's tricky to see how much someone is shedding before you know that they were infected, right? So that data is very sparse, and it's one of the biggest gaps that I think we have right now is getting more good shedding data on kind of a wide range of diseases.
Presumably you'd have to like prospectively enroll.
Yeah.
Okay.
Exactly, and I mean, we've talked about that. I wrote a whole grant that then CDC canceled the whole funding round. I'm gonna find somewhere else to submit it, but we had this whole project where we were gonna just like regularly take samples from people and assume that some percentage of them, families with young kids, are gonna get infected with, flu, RSV, COVID every year. And, get some of those samples that might be before people are infected, or somebody's got the flu, you take samples from the rest of their family until the rest of them have the flu, because it's very, very likely, right? I mean, we don't want them to get the
Sure.
flu, like,
But given that it's gonna happen.
But if you're gonna, let us take some stool samples before you do, so that kind of study, we need more of. But it's very tricky. There are human challenge studies of some, pathogens. So like, there's one at Emory that's a flu human challenge study. And so you can get information on that because you know exactly when you've infected somebody, and then you follow them and see how much they're shedding. So doing more things like that will be really helpful. But the early warning really, really depends, not necessarily so much on, do you start shedding a day or two before you have symptoms, but it has to do with how quickly can we test the sample and report the data. Because a lot of the delay in clinical data comes down to compiling it and reporting it. And so, we want to know more information about shedding because it will improve our ability to model lots of things off of the wastewater data. But in terms of our speed of information and response, simply being able to say, on this day, everyone was shedding it, and we processed the sample the next day and then reported it the next day. That is very fast compared to a lot of our systems that have to bring data together that's often voluntarily reported from complex disconnected systems. Yeah. We're about out of time, but if there's one more question, or I don't know, Natalie, you tell me.